Professor Pitchappan, who most of you I'm sure will know. Professor Pitchappan is a pioneer and national leader in immunogenetics, immunology of infectious diseases, transplantation immunology, and human genomics in India. Professor Pitchappan's research has received genomic predisposition to leprosy, genetic susceptibility to tuberculosis, and HLA diversity in Indian populations, including the identification of descendants of the first out-of-Africa migration to India around 60,000 years ago. Professor Pitchappan has authored more than 120 refereed scientific publications and has participated in numerous international congresses. In 2008, Professor Pitchappan was invited as one of only 16 speakers worldwide and the sole representative from India at the UNESCO conference, the first Great Migrations of Peoples in Paris. During and after tenure as professor at Madurai Kamaraj University, Professor Pitchappan has led multiple Indian-focused research projects and served as director or advisor to institutions and agencies, including the Indian Council of Medical Research, Andaman and Nicobar Administration, Drug Controller of India, Nilgiri Adivasi Welfare Association, Chettinad Academy of Medical Sciences, UGC, EMRC, and the Global Genomic Project. Professor Pitchappan currently advises the Ancient DNA program of the government of Tamil Nadu and Madurai Kamaraj University. Professor Pitchappan was elected Fellow of the National Academy of Medical Sciences, New Delhi, and the Indian Academy of Sciences in Bengaluru in 2002, and has received several Lifetime Achievement Awards, including from the Genome Medicine Consortium of the United Kingdom. He's a strong advocate of public engagement with science, and he's pioneered HLA transplantation matching services for kidney and bone marrow transplantation at Madurai Kamaraj University. Is now widely used by CMC, Vellore and transplant centres across the Deccan region. He's contributed to international documentaries such as the famous Story of India, BBC series, Journal of Man, the National Geographic series, bringing complex science to wider audiences. In 2023, Professor Pitchappan established the Pitch Foundation to improve education and promote appropriate green technologies, and continues to work towards a vision of quality India. So it's with great pleasure that I asked Professor Pitchappan to come to the stage and give us his presentation.
Thank you, Professor Benjamin, for the introduction. This interdisciplinary approach and the human biology oriented, we started some 40 years ago. I met Professor V.I. Subramaniam in the International School of Dravidian Linguistics with some of my data, and he said the answer is in DNA. Now, I will just simply go through one after another the slides and try to stick to the time. The question is whether language is a prelude or language as a prelude is a question to the cultural evolution. We all know that the culture, but the language development still is highly debated. We use the DNA technology. You know, simple mouthwash. In just 1 hour, I can collect the DNA of all the people in this room just by giving 1 ounce of water, ask you to spit it to a cup. Another 1 hour, we just spin and store it. That's all. We have got your whole genome. Okay? So that kind of technology you should develop. Ancient DNA aims at that. Now, I deal with here the male migration. Okay? The males have a chromosome called non-recombinant Y region in the Y chromosome, which doesn't easily recombine. So it is inherited from father to son and son to grandson and so on. It is not inherited by daughters. Okay? This is responsible for the maleness. Now, man originated in Africa some 200,000 years ago. Some 70,000 years ago, due to some calamities, a small band of people left Africa and traveled through the coast of India, reached Australia in another 10,000 years. This is called first out of Africa migration. If we just consider the route, the pointer doesn't work? Okay, A as the root, A, B and C, these are all single mutations. Pointer? Not change, pointer I want. Not working, okay. All right. So A, B, C are all the base of the trunk of the tree. If you consider the mankind male lineages as a tree, and then F, H and L branches are mostly specific to India, whereas J, O and R, they are all from different regions, Central Asia, Oriental region and Eastern region. They all came over later to India. Okay? So these are all simple point mutations we detect in the DNA. Right, what is the result? So these are the methods that we use. Some 12,000 samples we collected from all over India, most of the states, and then genotyping was done using the state-of-the-art technologies, DNA technologies, everything in Madurai. And then appropriate statistics were also applied, and you can date, like your carbon dating, based on these mutations at the population level. We can tell the time of origin of that particular population. Okay? Now, what is the result? Just, this is the pan-India. Okay? Ancestry, you can call it. All the 12,000 people put together. Now, R1A, yellow color, represent 19.38% of the sample that we collected. And H1A, 19.3% again. Whereas O2A, 8.12%, R2A, 7.31%, F, 6.09%. Each one of these alphabet, as I mentioned, is the branch. Okay? Then Tibeto-Burmese O3A, Dravidian L1A. What do you mean by Dravidian or Indo-Aryan or Austroasiatic? Is in the principal component analysis, when you do that, there is a great correlate with these language speakers as well as the particular Y chromosome. Okay? That's what there is correlate. Then we asked a question, can we trace the language subfamilies from this? So this is just the language subfamilies, not this, that is the African AF, AA, Austroasiatic Kasip, and then Austroasiatic Munda, then Sino-Tibetan, Tibeto-Burmese speakers, Dravidian, Central Dravidian, Dravidian, South Dravidian, and so on. Most of the languages, you know that. Okay? Similarly, Indo-Aryan also, Magadhi, Maha, and so on. So when you did that, this is on the x-axis, I have given the various branches of the NRY, and then on the y-axis, I have given the various language families, and various factors also I just deal with. And Mike. That is the subfamilies of SD1 is South Dravidian 2, SD1 is S Dravidian 1, CA means caste, BR means Brahmin. Okay? The first column is the number of samples that we have collected. Now, look at that. I have made it as a histogram itself, here itself in the frequency table, and you have the highest frequency over here. What is important is this H1A is present almost in every Dravidian speaker. I am not presenting you the Indo-Aryan speakers here. So only Dravidian speaker. Whereas some others, this O2A is seen, this is actually Austroasiatic speaker, but seen in some of the Dravidian speakers. especially the North Dravidian speakers. That is actually gene pool has replaced in a language speaker. Then there is another specific thing, this one L1A, which we call as a marker for Dravidian, is mostly present in higher proportions, 0.13 and 0.14 frequency in South Dravidian 2 and South Dravidian 1 speaker. Okay? So it is not uniform, even among the language speaker as well as among the tribe caste continuum. If you just look at that, tribe, then caste, then the Brahmin. Okay? Now, to correlate it further, we made a contour map, elevation maps, and on the x-axis it is various subgroups of language speakers, and on the y-axis it is the various branches of the tree, and these are all, we can call it as the original Indian clade, which I showed you in the first tree diagram, and these are all the Neolithic. This, of course, very prominent only, this red one is 0.4, and then this yellow one is 0.2. Now, when we look at that, the H1A is present almost throughout India, as well as R1A or R clades. Okay, these two are very common. Please recall the first proportion diagram that I showed you, wherein the R1A and H1A were the biggest block of 19%. Okay? So you can imagine the frequency. More the frequency, more the people probably that branch established very well, and maybe natural selection or whatever be that the reason, they expanded. At the end, I will correlate this to sociology also. Now, in this diagram, I have put the root of the tree on the top and the latest one at the bottom. Okay, it is an inversion tree. But this, I inverted that. Then you look at that, so beautifully it comes up. Now, most of these are seen in the Indo-Aryan Brahmin people R1A. Okay? Whether it is South Dravidian or North Dravidian, everything is the same. And this is from the tribal, they become caste, and they also go in and integrate into Brahmin population at 10% level. Okay? So we are further working on the sequences to prove this point. All right? So this is typically the Austroasiatic and Mundari speakers or Tibeto-Burman speakers on the eastern India. So originally it looks like all the language speakers started as a tribal, including Indo-Aryans. They are called as Vedic tribes. Okay? Then they evolve, maybe culturally, as you talk, become a caste, and some people take over some Vedic philosophy, different directions, and Brahmin is a conglomerate of a functional group, rather. Okay? So we will see that later. So this is actually the forest plot of these NRY haplogroups, their fidelity as odds ratio, a log scale for haplogroup enrichment comparing the Dravidian and Indo-Aryan language data. You just look at that. F star, whichever I showed as a pyramid, they are all significant, and these are all the error margin, and these are all mostly in Dravidian, these are all in Indo-Aryan speakers. Okay? Now, then, this is what again? A picturesque representation for you to know These are all the different branches of the NRY tree. These are all the tribe. Look at that, this is the tribe. OTA is only in the tribe. Okay? And then North Dravidian and South Dravidian it is seen. Okay, and then where it is normally distributed also have been given. So, first, when the out of Africa migration man came into India, he starts at Western Ghats as tribe, represented even today at the highest frequency in the Sangam literature mentioned Paliyar, Pulayar, Kadar, Irular. They are the original people. 2024, Priya Murjani has confirmed this, and 50,000 years ago, that is the age based on the whole genome. Then you look at this specifically. This is a caste of Dravidian speakers, L1A2. Okay? Then the Brahmin group, they have the highest frequency at the bottom, R1A 576 and then M124. So, in other words, later arrivals came and overlaid like a layered cake. So what is the age of these? So we can, based on the diversity, STR diversity of these chromosomes that we studied, we can always calculate the age. This is what in the world literature gives 48,800. That is the F star, which we mentioned in the Paliyar, Pulayar, you know. It is the founder of India in the Western Ghats. Okay? Then in our pan-Indian data, it is 31,000. And when we consider as an individual population, a combination of 19 population or so, the mean is about 16,000. So read for yourself. This is a H1. This is a pan-Indian. Whether it was the original Dravidian cultural element is a question. We feel so. Then these two. Then comes the Southern Dravidian. This is not seen very much in the Central Dravidian. It is absent really, this branch. Central Dravidian, they have only these two. Then is this is the Vedic people or Brahmin. Now, your big question is, who are these people? I tell you, in Indo-Aryan language speakers, only 1/3 of the population belong to Brahmin. Okay? R1A also, only 1/3 belong to the Brahmin group. Okay. Now, once again, regarding the dating, people have argued, usually with younger clades, the calculation method bloats the age a bit. Whereas with the older clades, it suppresses a bit. So, in fact, David Drake and all other publications say about the ancient cDNA of the Indus periphery samples, about 4,000 years or so. So, yes. So, in summary, there is a layered paternal history. I will just read through of Indian population. This is based on the Y chromosome. It should be true with whole genome also, but we need to look at the female gene pool also. Female gene pool were all derived from the first out of Africa migration. Whereas as I showed here, male migrations have occurred time and again, and they form most of the northern Indian plains nowadays occupying. So there is a Y chromosome haplogroup which shows a continuous multi-layered paternal history, and there is also an indigenous Mesolithic foundation, as I showed you with the H1, L1A and F, dominating the tribal and Dravidian groups. And there is a continuity without large-scale male replacement in these populations. This is still about 20,000 years earlier. But with the Neolithic demic diffusion, the agricultural spread resulted in admixture, and this is very much with Austroasiatic and Indo-Aryan language speakers. Then the Bronze Age steppe, commonly they used to call it, probably that culture came in, brought in by a few people, but they are a small band of people probably, and they are represented by these haplogroups. But these haplogroups had a huge expansion only in India. This is in Tajikistan. That's the latest branch of RNA. They are not seen very much. They are there in the Europe, but in very low frequency. So, what is the take-home message that I want to tell? Synthesis. I am going to draw some references from literature for you to appreciate the value of this. So, the time scale overlaps with the late Paleolithic and Neolithic cultural evolution, whatever you are talking about. Maybe 10,000 years, maximum 12,000 to 15,000. Now, is it the culture that gave rise to the language or the other way? Right? What was the driving force behind such socio-cultural evolution? Whether it is the food, or the technology, or the agriculture, language, culture, society, whatever you call it. Was the chick first or the egg first? This is a common question that we used to ask. Okay? Now, look at that. This is a bit confusing, a large comprehension of India. This is actually the population size. And as per the population size, it has been started. This is from our literatures, census data, 2011 if I remember correct. Uttar Pradesh is the largest state, 16.51% of the population of India. And also with 15.1% of the land mass. Okay? And in fact, the production, if you look at also, it just gives the square kilometers and then population in square kilometer. But GST revenue, if you look at, it is very limited. Whereas in terms of agricultural produce, sorry, this is agricultural produce, it is actually comparable to that. So, what is the correlation? Then the whole of the northern belt, it is the Indo-Gangetic dome, which really is supported the huge paddy cultivation. That is how the population has expanded so much. And, okay, so it is actually the gene which led to language, in my opinion, and then the migration. And the food security and socio-culture resulted in the picture that we saw there. And for your information, there are genes coding for language, cognition, and everything. And it is this, FOXP2 language gene. Then executive functions, there are a few genes. Then there are multiple genes, and it is not controlled by only one gene. Brain development, neural connection, cognitive process, everything is determined by gene, whether you like it or not. Now, this person, in fact, Michael St. Clair, we invited, but he could not come. Maybe in the next meeting he would talk to you. He has published a monograph on this language, prehistory of language from perspective of Y chromosome. So he says a triangulated Y chromosome based modeling of language prehistory yielded desperately needed models of language prehistory, and it is highly reliable. So this is actually Pagel and Atkinson 2013 paper, wherein they put the Proto-Dravidian language as 13,500 years old, and it is entirely separate from all the rest of the language families, as evidenced by the grammar itself. And this is our study in 2012 showing the seven separate groups of Tamil Nadu. 7,000 years ago itself this social stratification has occurred. During the past 3,000 years, it did not intermixed. This is the same data. This is a Kolipakam 2018 paper. The modern sample, the age is about 4,500. Now, Y chromosome, once again, is a blueprint of men's health also. So, some common European lineage, HGI, has some increased risk of even coronary diseases. So there is a correlation between all this, whatever we are talking about. I hope that I have given you the information. So it is something like a chick looking at the world as it is hatched. We all need to relook archaeology, I wish. Thank you. Sorry, 22 minutes.
Fantastic. He kept to time pretty much. So that means that we have 5 minutes for questions and comments. I'm sure there will be a lot. A difficult thing to talk about. Or maybe I will kick off. So when you're talking about language and being part of the picture here in India, I mean, obviously language developed outside of India. And probably the first languages were probably the Khoisan languages, for example, in southern Africa, which are click-based. So in terms of whether genes or something within the body represents the origins of language and culture, I mean, I've always thought the development of the voice box is an absolutely crucial moment in modern human development of language. But it has recently been pointed out that you could have fully complex languages using clicks, so that the Khoisan have 6 clicks. And you can create a language without, in fact, having a voice box. So language could be much earlier, in fact, than we even originally thought. And so I'm wondering if really the genes hold
very old.
I wonder if the genes hold the key to language. But it's an interesting topic for debate.
Okay, thank you. Okay, thank you.
Why there is only one caste is mentioned, or like one particular region is mentioned? Are we tracing certain languages or tracing certain people?
Can you repeat?
So I see one particular caste is mentioned in the analysis. So because the focus is on tracing of that group of people, or like, yeah, I'm asking if it is focus is on that particular caste.
Brahmanism is not individual.
Why?
We do have the name of the caste and everything individually. You see, one is tribe, agree? The next is little evolved status. Then you can call it the caste. Can you? See, the Brahmin I mentioned as a functional unit of conglomerate. You know, among the Brahmins, some people have classified it as a preacher. Do you agree on that? They are in every vocation, you know. So, this is just, we could find a difference, we show. You can call it caste X and caste Y, I could have called it. And among the caste itself, we can stratify. That's what in one of the slides, 7 subsets I showed. We do have data on that. But this is overall generalization. there was a difference. We can consider it together, but there was a difference. Difference was so clear.
Actually,
Yes, culturally they are distinct. Do you agree or not? Yeah, that culture, you take it in the analysis part of it as a...
So you have a preconceived idea like Brahmins on the top?
No, I never said top. I never said top.
Because you say tribe, then caste, then what?
No, that's what the Y chromosome shows, the evolution. The youngest tree on the top is from the Central Asian, Indo-Aryan tree.
So you are not talking vertically but horizontally, right? Tribes, then caste, then Brahmins, or what? This is the question. But it has become very clear.
No, we will discuss about it. I will
actually,
explain to you.
another one question, sir. Actually, you are focusing in migrated Indo-Aryan group of peoples like that Brahmins or native peoples of Brahmin.
First to define. That.
Actually, in the local preacher, local native peoples also, some of the preachers also available. That is
That's
also
what
calling
I
as
have
a
shown them.
Brahmin.
I have
Indo-Aryan migrated peoples also
No.
called as a Brahmin.
Y chromosome, which is R1A1. That's what I showed. See, when you talk about culture, cultural disparities and differences, you need to consider. See, the Brahmins of Bengal eats fish. Southern India, no. The Brahmins here marry uncle niece. North India, it is a taboo. These are all the culture. See, when we had the tribals cremating, Kannada speaking, and the others, we could very clear difference. That's what I showed you. Time didn't permit me to explain that. Thank you.